Okay guys, welcome to the video again. So today we're going to talk about autosomal linkage, and there's two main types of autosomal linkage. One is complete and one is incomplete. So when two different genes are linked on the same chromosome, okay, that really means that the locus for each gene is actually on the same physical piece of DNA. So what we're going to do is start with complete linkage. Now, imagine we had an autosome chromosome. That's just any chromosome that isn't a sex chromosome. So it could be, for humans, chromosome 1 or chromosome 6 or chromosome 9. And we're going to start with the parents, and it's going to be heterozygous for two different genes. Now, I'm just going to use gene A and gene B. So it's a diploid organism with two sets of chromosomes. So for the first gene, gene A, there's going to be two alleles, two A alleles. And for the second gene, because it's a dihybrid, there's going to be two B alleles. So I'm going to go heterozygous for gene A, and the locus for gene A is going to be on this chromosome here, of which there's two copies because it's a diploid organism. And the second gene in our dihybrid is going to be gene B, and that's going to be a different locus to gene A, but it's going to be on the same chromosome. And they're heterozygous for both genes. So one dominant allele and one recessive allele for each gene. So if we have a look at gene A then, so we've got two chromosomes, and we're going to put this locus on the first chromosome. It's going to be our dominant A allele, which is that one. And on the second matching homologous chromosome at the same locus, so hopefully we can actually see the locus is at the same position here, we're going to have the other allele, which is the recessive allele. So it's a diploid organism with two sets of chromosomes. There's two alleles, therefore, for gene A. There's a dominant allele and a recessive allele on the homologous chromosome. Now, because gene B is linked, we're going to do a different locus, but it's going to be on the same physical piece of DNA. So our dominant B allele is going to be linked on the same physical piece of DNA as the dominant A. And the second allele for gene B is going to be at the same locus, but on the homologous chromosome again. So the genotype of this parent is big A, little a, heterozygous for A, big B, little b, so one dominant B, one recessive B, that's heterozygous for gene B. So this is what it would look like. However, obviously S phase of interphase, we know the DNA does replicate, and that's semi-conservative replication. So what I'm going to show now is the DNA, how it would look after S phase of interphase when we've had replication of the DNA. So this is one chromosome, it's one molecule of DNA, but we know semi-conservative replication is going to make an identical copy of that piece of DNA. And this is now our replicated chromosome, and this was S phase, so I could just put S phase here of interphase. And don't forget, sister chromatids are genetically absolutely identical. So this replicated sister chromatid will have a dominant A, and don't forget this second locus for the second gene, it will have dominant B. Now the second chromosome, that's also going to replicate its DNA. So we'll just do the identical sister chromatid here. Don't forget the centromere links the two sister chromatids. And on the second homologous chromosome then, we'll just put in the sister chromatid on this side. So we can see after S phase, we've got the two replicated homologous chromosomes, and we know this chromatid, sister chromatid on the left, will be in one gamete. This identical sister chromatid on the right here will be in the second gamete. This sister chromatid of this homologous chromosome will be in the third gamete, and this sister chromatid, which is identical to this one on my left here, will be in the fourth gamete. So the gametes you get for complete linkage would quite simply be dominant A and dominant B, and little a and little b. So recessive A and recessive B, and that's it. There's only two types of gametes in this case, this dihybrid. So we get gamete A, second gamete here, third gamete here, and fourth gamete here. So you can see there's only two types of gametes. So this would be complete linkage. These would be the gametes that would be made through that. So if we took that parent and then we crossed it with a second parent who has the identical genotype, the first parent would make these two gametes, and the second parent would make exactly the same gametes because it's got the same genotype. So if we did a quick Punnett square, what you'd find is that the gametes from the first parent could fertilise the gametes from the second parent. So simple Punnett square then. So this is parent 1 and this is parent 2 at the top. So we're going to recombine all of the alleles here. So it would be that genotype for one offspring. It would be that genotype for the second offspring. It would be that genotype for the third offspring. And it would be, watch this, that genotype for the fourth offspring. So what you'd find if you had two heterozygous parents for both genes and you crossed them, in theory anyway, we would have dominant A, dominant B phenotype, dominant A, dominant B phenotype, because remember one dominant allele gives a dominant phenotype. There would be a third dominant A, dominant B phenotype, and the fourth offspring would have recessive A, recessive B phenotype. So if we have a look at complete linkage then, and we said, well, what phenotypes would we expect from this cross? Remember, the parents are this. We'd have dominant A and dominant B phenotype. And that would be 3 out of every 4, because remember, our ratio is 3 dominant A and dominant B to 1 recessive A, recessive B. Would there be any dominant A and recessive B phenotypes? Well, actually, no, there would not. Would there be any recessive A and dominant B phenotypes? Well, no, there wouldn't. Would there be the recessive A and the recessive B? Yes, there would, because we've just said that 1 of every 4 offspring would have that phenotype. So if we had 1,600 plants, for example, that we made, we would expect a 3-to-1 ratio of the double dominant phenotype to the double recessive phenotype. So if you divide 1,600 by 4, you're going to get 400. So 3 times 400, we would expect to see, of these plants, 1,200 with the double dominant and only 400 with the double recessive. So that'd be complete linkage, and it'd be a 3-to-1 ratio of the phenotypes you can see hopefully here on the left. So this would be a genetic cross producing 1,600 plants, which is quite a big number, but hopefully you can see how it's split in the ratio. So I'll put here numbers. Now, if we have a look at incomplete linkage then, so this time we're going to go the same genotype for the parent. So I'm going to go this genotype again for the parent. It's exactly what we had at the top, but this time I'm going to put the genes here. So that's the dominant A allele, and here I'm going to have my dominant B, and on the other homologous chromosome, don't forget we've got our recessive A, and here we've got our recessive B. But of course we get S phase of interphase, so we just replicate the DNA molecule to make the 2 identical sister chromatids. That's S phase. Okay, now what I'd like to do is on the sister chromatid the identical sister chromatid to the one on my left here of this homologous chromosome. I am going to put in the dominant A here, and on the other sister chromatid of the other homologous chromosome, I'm going to put in my recessive B. Now, this one here, again, would be the same as the sister chromatid, but I'm going to have crossing over take place now. So this is where incomplete differs to complete linkage. So I'm going to say my chiasmata point for crossing over is here. So the crossing over point is the chiasmata point, and this little chunk of chromatid is going to physically swap with an equal length of chromatid from the other homologous chromosome. Now, this chromatid here is not identical to this chromatid here because obviously they do have different alleles. So these are not sister chromatids, they're just different chromatids, but the chromosomes here, the replicated chromosomes, are homologous. So we would say this is the chiasmata point. So this chunk here, hopefully you can see that, is going to swap over with this equal length of chromatid from the other homologous chromosome. So the lowercase recessive B allele on the right here is going to swap over with the dominant B allele that was originally on the chromatid on the left on the other homologous chromosome. So what you end up with, if I just redraw this here, is, I'll try and do it a little bit smaller. Get the centromeres. So the dominant A allele at the top, dominant A there, recessive A here, recessive A here, dominant B at the bottom, but this time I'm going to swap it over. So here I've got the recessive B on this sister chromatid, and here, because remember we've swapped over the alleles, is the dominant B, and here the original recessive B. So remember, this chromatid ends up in gamete number 1, this sister chromatid ends up in gamete number 2, this chromatid of the other homologous chromosome ends up in gamete 3, and this sister chromatid to this one, the third one here, would end up in gamete 4. So the gametes you get from crossing over, which we know is quite rare, would actually be, and I can put these at the bottom now, dominant A, dominant B. Okay, don't forget big A, little b. This is the second gamete over here. So it would be dominant A, recessive B, recessive A, dominant B. So recessive A, dominant B. And don't forget recessive A, recessive B. So this would be incomplete linkage. Now, you do get the 4 different gametes with incomplete linkage. And these would look very similar to the gametes that you get from just regular Mendelian inheritance, where the 2 genes are on different chromosomes. However, the crossing over points, the chiasmata point, where we swap over the 2 alleles from the chromatids, this is quite rare, this process of crossing over, and it happens in prophase 1. So I'm going to put here, crossing over happens in prophase 1 of meiosis 1. So imagine 10% of cells, maybe the germline cells that undergo meiosis, 10% have crossing over. That means 90% wouldn't have crossing over. So if we think about what gametes are being made, well, 10% of cells would give rise to these 4 gametes, but 90% of cells with no crossing over, because we know crossing over is rare, would give rise to these gametes. Dominant A, dominant B, recessive A, recessive B. Now this is very similar to the first case we had at the top with no crossing over. So crossing over, because it's rare, it happens in about 10% of cells, germline cells that undergo meiosis 1. You do get the 4 gametes, including the 2 recombinant gametes. But remember, 90% or so of cells wouldn't have crossing over because it's quite rare. So you get big A, big B, recessive A, recessive B is the main gametes. So when we look at incomplete linkage, what we're going to find is that we don't have an even number of the 4 different gametes. We by far have more of this gamete, by far, and we have more of that gamete, by far. And we have very few numbers of the recombinant gametes, which are this one and this one. So most of the gametes that are going to fuse together include big A, big B, or little a, little b. There's a small number of recombinant gametes, which would give rise to the recombinant phenotypes. So if big A, big B fused with big A, big B, you'd get homozygous dominant A, homozygous dominant B, so you'd have the double dominant phenotype. If little a, little b fused with little a, little b, you'd get two little a's and two little b's, you'd have the double recessive phenotype. And you get the recombinant phenotypes by having the smaller number of recombinant gametes fuse with the two gametes at the top. So if you get a really weird ratio, it's incomplete linkage, where the two genes are on the same chromosome, but you get rare crossing over that occurs between them. The further apart the two loci are for each gene, A and B, the more likely chiasmata will form actually between the two genes. So let's have a look at the offspring we might expect from this incomplete linkage then. So the phenotypes would be the same as that list at the top. So I'm going to say dominant A and dominant B phenotype. I'm going to say dominant A and recessive B as the phenotype. And then the other recombinant phenotype, so recessive A and dominant B. And the fourth phenotype would be the recessive A phenotype combined with the recessive B. That's the double recessive phenotype. So again, if we had 1,600 plants or offspring, we might expect a bit of a weird ratio. So it might be that we have 900 of the dominant A and the dominant B phenotype, and 500 of the recessive A, recessive B phenotype, because remember, we by far got more of that and that gamete, which can fuse together. So we could have two of these fused together, we could have two of these fused together, or some of these could fuse together, which again would make the phenotype at the top. We'd have very low numbers of these dominant A, recessive B phenotypes and recessive A, dominant B phenotypes. So this ratio you might see as something like 9 to 1 to 1 to 5. And you can see the two phenotypes in the middle here would be in very low numbers in respect to the double dominant phenotype and the double recessive phenotype. So we do still get all four phenotypes here, but in a bit of an odd ratio. Now, if we have a look at Mendelian inheritance then at the top, so this is the one that you would have started with the dihybrid. We'll do the same genotype at the top. So we'll go heterozygous for both. But we know these parents, if you cross two of them, they both make gametes. Big A, big B, big A, little b, little a, big B, and little a, little b. And they make them in about equal numbers. So that would be the first parent, and then this would be the second parent. Imagine we had the same genotype here. So again, it would be big A, big B, big A, little b, little a, big B, and little a, little b. Now, if you did this Punnett square, you get 16 offspring in theory. These are the expected offspring in your Punnett square. And you'd get the classic, in this case, 9 to 3 to 3 to 1 ratio. So the 9 would be the double dominant. So dominant A, dominant B phenotype. The 1 of the 16 would be the double recessive phenotype. And then you get the two recombinant phenotypes in about 3 of every 16 for both. So let's just fill in this table here. This is for Mendelian, where there's no linkage. So here, gene A would be on one chromosome, and gene B, the locus, would be on a completely different chromosome, and they wouldn't be linked together on the same piece of DNA. So we can add this in. So the phenotypes then that we've seen. So dominant A and dominant B. Okay, so again, imagine we had 1,600 plants or offspring. So 9 over 16 times 1,600. will actually give you 900 offspring. The dominant A, recessive B, recombinant phenotype would be 3 of every 16. So that'd be 3 over 16 times 1600, and that will give you 300. So hopefully you can see the classic 9:3 to 3:1 ratio. The recessive A with the dominant B, again, that would be the same. And lastly, the recessive A with the recessive B phenotype, remember phenotype, not genotype, would be 100 of every 1600, or 1 over 16. So our numbers would be classic Mendelian if there was no linkage. If there is complete linkage, you expect a 3:1 ratio of dominant A, dominant B to recessive A, recessive B. And please don't forget that that would be with a heterozygous parent for both genes, as you can see up here. And then with incomplete linkage, you'd get an odd ratio with by far higher numbers of the double dominant phenotype and the double recessive phenotype, with smaller numbers of the 2 recombinant phenotypes, as hopefully you can see here. So any odd ratio that does not meet the Mendelian ratio that you might expect, you're probably going to be thinking about, if you get 4 phenotypes, incomplete linkage, which is this one.